Thank you all for joining. I'm so happy to be here. My name is Brittany Anderton. I am Associate Director of Research Talks at iBiology. And for those of you that might not know what iBiology is, it's a small nonprofit organization based in San Francisco. And what we do is we make free online videos for sharing the wonder of research, largely with the scientific community, but also for those who are science enthusiasts. So we welcome you all here today or tonight, or wherever you are in the world. And we're so happy to be here to celebrate science with you all. I'll let Dipti, my speaker, introduce herself as well.
Hello, everyone. I'm Dipti Nayak. I'm an assistant professor in the molecular and cell biology department at UC Berkeley, where I've been for the last year or so. I look forward to interacting with many of you via chat or other features that we have today, and to watching this video together with all of you as well.
Excellent. Yeah, we're happy to share our experience on working on this video with all of you and field your questions. So just a couple of ground rules before we get started. I think maybe some of you are familiar with the webinar function in Zoom. The chat function, let's have that be side chats. I won't be monitoring that, nor will Dipti, so feel free for any of you in the audience to chat with each other on that. If you have questions for us that you'd like us to answer after the video, send those in the Q&A, and my colleague Kevin McLean will be fielding those to us. And so Kevin, do you want to pop in and say hi? Because people might hear your voice later.
Yep, I'm a real human person. I'm here. Yeah, so if you do have questions along the way, I'll be monitoring that Q&A box, and I can pass those questions on to Brittany and to Dipty.
Great, thanks, Kevin. Yeah, so there is a man behind the voice. Yeah, so we will be fielding your questions after the video, but we thought we'd start with a little bit of a preamble and introduction to this project. And I think what we can start with is a background of how this came to be and how Dipti and I came to know each other and our experience during the recording. So in fact, we just released this video within the last month, but we recorded it in December of 2019. So it's been many months in the making. And Dipti, I don't know if you recall that day or what your experience was being in the camera for so long.
Yeah, I know it. Yeah, it was December. It was somewhere in mid-December, which feels like a lifetime ago right now with everything that's happened in between. But what was it like? I think it was my first time ever being in front of a camera, so I didn't quite know what to expect when I got there. I must say though, working with all of you was great, because I think the moment I got there, I kind of realized what I was walking into. So there was like a moment of anxiety that, oh, I'm not prepared for this, but just then kind of being walked through the process and realizing that at the end of the day, this was just another conversation, and a conversation about things that I care deeply about, and that have kind of motivated my academic passions for the last decade or so. It became relatively easy and straightforward, and I'm thrilled by the outcome. It's beyond what I could have imagined it would be.
Yeah, that's great to hear. I really enjoyed working with you, and a little bit of background for those of you that aren't really familiar with this project. So we at iBiology set out to at least create one video, and it's turning into a series of videos now where we are illuminating this mysterious microbe known as Archaea, which is the organism that Dipty's lab focuses on at UC Berkeley. And through some connections we had, we learned about this really stellar scientist who is excited about her research and can communicate it really well. And that kind of led to this event last December where we got together in a room for half a day, and I was peppering Dipty with questions for all those hours, and she was very resilient, very, very enthusiastic. And my role in this whole project has been as really a producer. I have a PhD in biology. I have dabbled in science communication and science education over the last, let's say, half decade or so. And in my role at iBiology for the last 2 years, I've been transitioning into a producer role where I've been thinking about how to use my skill set to create videos that bridge science communication and science education. And this was my first experience to take that skill set and apply it to a project on a topic that I know nothing about. I have no background in microbiology, but really, Dipti, you made the experience really fun and easy, and those interviews were engaging. I learned just as much, I think, as you all will learn from the video just in those few hours. So let's pause for a moment. We have some pre-video questions we want to ask the audience. I'll launch the poll here. We'd love to know who you are and what brought you here. So I see some responses coming in. And it looks like there's about 20 or so people in the audience. So I hope you'll, okay, many more are coming in now. So 80% of respondents are looking like they've heard of Archaea. And what brought you here to this event? A little over half of you want to learn more about microbiology. A quarter of you want to hear from a real life scientist. So we've got that in store for you. Oh, those numbers are jumping up. About a third of you want to learn what Archaea are, so we've got that in store for you as well. I'm going to close the poll in about 10 seconds, so feel free to hurry up and have your responses, and then we'll share the full responses with everyone. Okay, I'm closing in 2, 1, 0. So let's see here, nearly 80% of you have heard of Archaea. 43% of you came here because you want to learn more about microbiology. 43% want to hear from real life scientists. So we're so excited to provide that with you. Okay, so I will stop sharing that and head back to this conversation. So a couple of things before we get to sharing the video. You know, as a producer, I had some goals in mind about what we wanted to accomplish. We wanted to create something that could be appropriate for those at an introductory level that really wanted to learn about this mysterious microbe, maybe coming at it with very little knowledge about it, and could walk away knowing about how they were discovered, what the actual techniques were that scientists used to discover them, and what they're telling us now about evolution and microbiology. And Dipti, I want to hear your thoughts really about, you know, what makes you drives your work every day and being an archaea biologist?
That's a great question. So I think one thing I want to preface is I think as scientists, we often kind of spend most of our day talking to other scientists. And what that means is that, you know, we can kind of think about some abstract problems about the specific systems that we study and then get really excited by them. The one thing that I think motivates me on a regular basis is why what I do matters beyond just me and the group of people like me who study the system, but also why on a much, much broader scale, like why should anyone be studying Archaea? And you know, I hope this comes across in the video, and if not, please feel free to ask questions afterwards. Like, why is this important? Why should we be funding these kinds of research, this kind of research? What can we learn and how can we apply that to society and the betterment of it? And you know, the organisms that I study specifically are involved in production of this greenhouse gas that contributes significantly to climate change, which is an extremely important problem. So thinking about the fact that we do really fundamental research that can eventually have a significant impact on society, but you know, maybe finding solutions to climate change is something that has started to motivate me a lot more since I've become a professor.
Yeah, and thank you for sharing that. So this video won't touch so much on the role of Archaea in climate change, but please stay tuned for another video that is in the works that is stemming from, again, that interview footage that we collected last December. And it's going to be focusing more on Dipti's research and having her tell about the role of Archaea in the climate change cycle and the methane cycle. And so if you'd like to stay tuned to be able to know when that video is released, I encourage you to go to iBiology's website and sign up for our newsletter. Our website is iBiology.org. And then you can just know when that video is going to be released. We're also on Facebook and we're on Twitter. So there's easy ways to follow us, and then you'll be in the know for when that's coming. It'll be coming out in the next couple of months. So I think without further ado, maybe we'll turn it over to watch the video, and I hope that will stimulate a lot of conversation so we can actually engage with you all. It's hard to know who's here and what your interests are, and so we want to be able to field those questions. Is there anything, Dipti, you want to share before we continue?
No, I can't wait to get started.
Great. So I am sharing in the chat right now a link to the video, but no need to click on that because I'm also going to be sharing my screen. If for whatever reason, internet connectivity or something, you're having trouble watching the video in the shared Zoom webinar, go ahead and click on that link and you can navigate to your a web browser where you can watch the video. It's about 12 minutes, and you can come back to this Zoom webinar and engage in the Q&A. But I will try right now to share it in this Zoom webinar, and we can all watch together. So Dipti and I will be turning off our, okay, so there's not a YouTube link. Oh, let me fix that to all panelists and attendees. Thank you all. Yes, thank you. So I think all panelists and attendees should have shared it. And Dipti and I will stop our videos and mute ourselves, and we'll get right to the video. So we hope you enjoy it.
I think Archaea represent, to some extent, a blind spot in our understanding of natural diversity. You hear a lot of people talk in many different contexts about diversity, and they'll bring up viruses, they'll bring up eukaryotic cells, and they'll bring up bacteria, but the archaea are often never even mentioned. What I want people to acknowledge when they hear about archaea is that this is a completely distinct domain of life that we should recognize for what it is, rather than blend it in with some other domain. Understanding the tree of life, trying to figure out what the evolutionary relationship of all life forms on our planet. You know, it's a point of curiosity. We all want to know where we came from. For the longest time, we've kind of had this belief in biology that there's these two different and extremely distinct forms of life on our planet. One is everything that we can see, you know, right from the insects to the trees, human beings, other mammals, fishes, and so on and so forth, you know, the eukaryotes. And then there's everything that's invisible, and they were called the microorganisms or prokaryotes. So prokaryote was this all-encompassing term that was used for unicellular microorganisms that did not have organelles within the cells. So their chromosome essentially just floats around in their cytosol. In contrast, the eukaryotes are organisms that have kind of evolved these complex organelles, one of which is the nucleus, which encapsulates all of their chromosomal material. And so that distinction was what was used to kind of make evolutionary relationships between everything that does have a nucleus and everything that doesn't. Turns out that it's not as simple as that anymore. In 1977, Carl Woese at the University of Illinois wanted to use not just how organisms look, their morphology, as a way to understand how they related to each other, but looking at parts of their genetic material, their DNA, to make these evolutionary connections. The evolution of new species begins with mutations in DNA. These mutations can change genes, which in turn can change the physical traits of organisms. Sometimes, when an organism passes these genetic changes to their offspring, it can cause a new species to emerge. In the 1960s, scientists began to wonder if they could determine the evolutionary relationship between different species by comparing their DNA sequences. The more similar the DNA sequences of two organisms, the more closely related they must be. Less shared DNA suggests a more distant relationship. To build a tree of life using this approach, Carl Woese needed to find a gene that he could compare across all life forms. He chose one that is needed for cells to perform one of their most essential functions, making proteins. By comparing the sequence of this gene between different types of organisms, he was able to infer the tree of life. This particular molecule that he narrowed in on is called the 16S ribosomal RNA. By looking at just this one particular molecule, he was able to glean kind of the evolutionary relationships of these organisms without even having to look at them. So the story goes that Woese was looking for microbial samples to do 16S ribosomal RNA sequencing for. He went up to one of his colleagues and asked him for these weird methane-producing microorganisms that his lab was studying.
This colleague was Ralph Wolff, who pioneered the study of these methane-producing microbes. And Ralph then handed him a vial of these methane producers and said, yeah, take these bacteria if you want to look at their 16S ribosomal RNA. And when Carl Woese and his students took a look at those samples, they found that they were nothing like any of the others that they'd looked at, to the point that I think they had to repeat the study at least 2 or 3 times to validate the fact that the 16S ribosomal RNA was in fact that different. And so when you look at them under a microscope, they might look similar, but if you look deeper into the cell, into the DNA, you see that they're actually a very different group of organisms. So he knew that he was onto something really, really big. The microbes that we all thought were just this one big pool of prokaryotes were not that. There were the bacteria that are very well established and studied, but then there were these archaea, this enigmatic third domain of life. Woese got so excited that as soon as he made that first 3-domain tree of life, he called up New York Times, and it became the front page article the next day. It was this interesting moment of serendipity. The reason why we discovered archaea was this person, Carl Woese, who had this pioneering technology, but also the fact that his lab was situated next to that of someone who was studying in archaea, and then it was those two things coming together that gave rise to this paradigm-shifting concept in biology. So archaea initially, when they were discovered, were found in extreme environments. So one of the first archaea that was sequenced was isolated from a hydrothermal vent at the bottom of the ocean. Now, we've essentially found archaea everywhere in the environment around us as well as within us. So there's archaea on our skin, there's archaea in our oral cavity, we have archaea in our guts, and every other place that you can think of, there are archaea. So often they're around us and we can even see them sometimes. We just don't know them for who they are. For instance, if you are flying into the San Francisco airport, you'll see these salt flats that have bright colors as you land. And that's just a visual cue to you saying, here are archaea. You don't have to go too far to find them. They're everywhere. So the methanogenic archaea that my lab studies, they produce 80% of the methane that's being released to the atmosphere annually, and which has a significant impact on climate change. They also have a very direct impact on human health sometimes, because these methanogens in our distal gut can be up to 10% of the microbial population, which is substantial. So the amount of calories that you get from your food, and also how you digest your food, what kinds of components come from it, some of those factors also depend on these archaea in your gut. These archaea are actually a lot like us. So the way the cells replicate, or the way one cell becomes 2 cells, the way the cells make their proteins, a lot of those things are very similar to the way we do that, compared to the bacteria that are also unicellular. And what that's leading to is this theory now that the last common ancestor to all modern-day eukaryotes came from an archaeon. There's kind of been a revolution in the field of archaea in the last 5 years. So people went and found these samples in different marine sediments, and when they sequenced the organisms that live there, they found these very evolutionarily distant group of archaea called the Asgard Archaea. So the Asgard essentially is a group. Within the Asgard, you have the Lokis, and then a bunch of other archaea that are named after Norse gods. These Asgard Archaea, they have proteins in their cell that are called eukaryotic signature proteins, or ESPs. And the reason why I'm highlighting this is because they're typically proteins that are only associated with eukaryotes. These ESPs have not been found in other archaea or in bacteria, but now we're seeing the presence of these proteins in archaea as well. So these organisms, when you put them back on the tree of life to figure out where they are, they've essentially changed the topology or the way the tree looks now. Previously, there were 3 domains, and there was a shared ancestor between the archaea and all of modern-day eukaryotes. There was a last common ancestor that we shared, and then the archaea split off and the eukaryotes split off. But now what we're seeing is that there is an archaeal ancestor that gave rise to the eukaryotes. what this seems to indicate is that these eukaryotes are branching from within the archaea and are closely related to these distant groups that we've recently found. So that essentially makes archaea even more closely related to us now than we'd initially believed. And that's kind of changing that view of the 3-domain tree of life back to maybe a 2-domain tree of life. But the 2 domains are different. The 2 domains are both actually unicellular organisms, and the eukaryotes are branching from one group of these unicellular organisms. So now that we're thinking about the tree of life slightly differently, one question that arises as a result is, how did that unicellular archaeal ancestor give rise to this complex eukaryotic cell? Now, one key evolutionary step that happened along the way to becoming a eukaryote was the fact that these cells had to acquire the mitochondria. What one hypothesis out there is that an archaeal cell engulfed a bacterial cell, a free-living bacterial cell, and eventually that particular bacterial cell living inside the archaeon went on to become the mitochondria as we know it today. So these loci archaea encode these eukaryotic signature proteins. And when we look a little closer into what these proteins do, they also provide us a hint as to how the complex eukaryotic cell may have arisen. A lot of these proteins encode for interesting cytoskeletal features, so features that make up the body of the cell. They produce these appendages outside the cell. And these appendages look like they could be used to engulf another organism that would eventually become the mitochondria for the eukaryotic cell. There are also proteins that confer the cell the ability to engulf other cells through endocytosis. All of these proteins have functions that we can use to kind of make sense of how that engulfment of a mitochondria may have occurred. It almost seems like these Asgard Archaea are primed for some of the functions that we ascribe to have happened for the first eukaryotic cell to have arisen. It's an interesting time to be an archaeal biologist because we're kind of in this major transition in thinking about archaea from an evolutionary standpoint. Right now, we're in this phase that we're gathering a lot of information, and that information is kind of letting us build new hypotheses about the tree of life. As more information is revealed to us, we'll kind of keep refining that tree of life. It is extremely likely that in the future we'll find something else, and that might make us want to revisit the tree of life again. There's such an understudied group of organisms that I don't think there's one right question to ask. I think there's so many things that we can look at. We know what's unique about eukaryotes, and similarly with the bacteria, but very little is known about what it means to be an archaea. What is kind of unique and distinctive about them? We have a few things that we know, but we don't really know their deep, dark secrets, so to say. It's a good time to go learn more about that.
Okay, so I hope everybody had a chance to watch it, and I hope the streaming worked out okay. I don't know if any questions have come in yet, Kevin.
Yeah, we've got a few questions coming in already. Let's see, maybe let's start with, there's a question from Blake who asked, does the discovery of Asgards and their connection to Eukarya overturn the idea of searching for a luca? So maybe Dipti, if you want to take that question and also maybe explain what some of those things mean.
Yeah, sure. Well, first, Blake, thank you for the question. And let's see. Yeah, first let me break down this acronym. What's luca? So luca is the last universal common ancestor, and that kind of builds on this hypothesis that all of life that we know exists on our planet today came from one ancestor, which was the last universal common ancestor. So life arose on our planet precisely once. And so this question pertains to this idea that we just recently in the archaeal world discovered this very distant group of archaea called the Asgard Archaea, and kind of are now learning about what their connection to eukaryotic cells is. Does this discovery have anything to do with the last universal common ancestor? And I would argue probably not. So, and the reason for that is that luca happened at the last universal common ancestor dates back to at least 4 billion years ago, if not more, definitely more. So, what happened that far back in time is really hard for us to infer today, especially for these microscopic organisms, but also that particular event predates all of the events that we're talking about in this video. So it doesn't really impact, the discovery of the Asgards. In a nutshell, the discovery of the Asgards doesn't impact the, how luca may have arisen. And that's a slightly different question that we can get into more if required.
Great.
Yeah, I think that is a great question. And in fact, we debuted this video via a YouTube premiere just a couple of weeks ago, and luca was coming up quite a bit. And I don't feel really equipped to answer that, but I think in the future, we will aim to create a video that can be linked to this type of material and put all this together for a teaching tool and clarify all of that.
I think we've got more questions. I'm gonna maybe stick with one that's on the same vein from Shannon. And she just asked, can you expand on the ESP evidence that connected archaea and eukaryotes? And how is that connection made?
Sure. Is it okay if I go first, Brittany?
Of course, you are very much the expert here.
Yeah. So just to reflect back on what the ESPs are. So these are eukaryotic signature proteins. And the reason why we call them that is because, as I mentioned in the video, all of our cells are eukaryotic cells. So when you look at those diagrams of what a cell looks like, the cell is packed with tiny little compartments called organelles. These organelles, the way they form inside a cell, require specific genes. And those genes are eukaryotic specific genes because only organisms that make all of these organelles need them. So when we typically thought of the prokaryote, so bacteria and archaea that don't have these compartments, so they don't have these organelles, we didn't see these genes required for the formation of those. So we kind of made sense of it based on that. But now, when people discovered the Asgard Archaea, they find that these genes that we typically thought were involved in eukaryotic specific functions related to organelles are also found in archaea. What they're doing in archaea, we're starting to just piece that together. There's some information out there on a few of these genes that form the cytoskeleton or the compartments of these cells themselves and influence how the cells divide. But there's a lot more that we need to learn. But the fact that these genes exist in archaea before the organelles may have arisen seem to kind of link the archaea to eukaryotes more closely, and that's the connection that the video refers to.
Great. Let's see. Brittany, did you want to add anything, or should we go on to the next?
I can't say I have anything to add because everything I know about ESPs I learned in the interview with you, Dipty, and so that's the extent of my knowledge at this point.
All right. Yeah, I'll definitely keep on asking questions in the Q&A box. I want to make sure I get to Esmeralda has been waiting very patiently. She asked her question before the video. She was curious about for both of you, if you could talk a little bit about your path into the careers you have right now, and maybe Dipti also touch on how you got interested in archaea in general.
Sure. So I've had a pretty meandering path to get to where I am for my undergrad, and I grew up in India, so I did my undergrad there. I studied environmental engineering, and I think what I was really fascinated by back then was this concept called bioremediation, which is taking plants or microbes and using them to figure out a way to clean up pollutants in different kind of ecosystems. So that's kind of where I started. Then I moved to the States as a graduate student, still in an engineering department, but soon kind of I got fascinated by the microbes more than the processes that they were conducting in the environment. And that kind of introduced me to Archaea because I was interested in metabolisms that were related to the environment. And as I briefly mentioned in the video, and even before the video was played, there's a strong connection between these archaea, these methanogens that I study, and climate change. And then kind of that's always been something very close to my professional career, is trying to find the nexus between biology and the environment. So that's kind of what's led me to studying archaea. So after grad school, I specifically went and worked in a lab at the University of Illinois, where these archaea were discovered, to learn more about them, to figure out how to manipulate their genomes, so to develop genetic tools for them. And that's what my lab does now. We take these genetic techniques. Some of you may have heard of CRISPR, which the technique that won the Nobel Prize a few weeks ago. So in our lab, we use CRISPR for archaea, and actually try to map what specific genes in these organisms do.
Yeah, and just another plug there for this upcoming video that's in the works. We walk you through, we'll be walking the viewers through how Dipty's lab uses CRISPR to manipulate the genomes of archaea and understand them better. So if you want to learn about CRISPR and you want to learn about how CRISPR can be used as a tool in archaea, again, find a way to follow us on one of our social media outlets, and we will definitely provide that information in the near future. So for me, my background, I grew up in a rural area of northwestern Pennsylvania up near Lake Erie, and I was always fascinated with the living world, but I didn't know much about what types of careers were out there for me. And I knew I wanted to study biology, so I went to college and studied biology, and I really was going down a path of career options that were familiar to me. So maybe a dentist or maybe a veterinarian. And it wasn't until college that I learned that people did scientific research for a living, and that just totally changed everything about how I envisioned my career and my path with science. And I had the fantastic opportunity to do 2 research internships, one at the University of Pittsburgh and one at the Institute Pasteur in Paris, and it just changed my total attitude about what it meant to be a scientist and what being a scientist and doing science was. And I knew I wanted to go to grad school. So I had the great fortune of going to grad school actually here in San Francisco. I went to UCSF and I studied cancer biology. Cancer is a disease that affected me indirectly just through many of my family members as I was a child. And it was something that I wanted to contribute to identify better understandings of cancer so we could potentially identify therapies for it. And so I did 6 years of research at UCSF for my PhD. And during that time, I just got really interested in how we can communicate about science with the public and how we can teach about science to those that maybe don't have as much expertise as us. So from the perspective of scientists, like how can we do it better? And that led me to UC Davis, where I did a postdoc in scientific teaching and scientific communication and asking questions about how we can do that better. And I was looking at a kind of a case study or model system of GMOs because, as we're all probably familiar, the GMO debate is pretty contentious, and there's actually a lot of research around how to teach and communicate about it. So I dabbled in that for a couple of years, and then that led me to my role at iBiology. And so I have expertise and experience in biology, in science communication, in science education. I taught at UC Davis and at Sac State. So I bring all these things together and think really strategically now about how can we create video resources and resources that go along with these videos that come from iBiology to further our goals in communicating and teaching about science. And I think this video is really kind of the epitome of the things that I would like to continue to create. And if you have feedback about how we can improve on this or new directions we can go in this, we definitely want to hear that from you all today.
Great. Sort of continuing along some of these lines a little bit, we have a question from Elliot who wants to know what each of you learned in the making of this video. Maybe we can reverse the order. Brittany, start again, and then we'll have Dipty go after.
Yeah. So prior to working at iBiology and with the team at iBiology, I had no experience in film, and I had very little experience with video for science communication. Even at the amateur level, I hadn't done much. And this for me was such a learning experience in how we can bring the tools of filmmaking to further our goals at the science communication and science education interface. And as you saw, it wasn't just an interview of Dipty, it was hearing Dipty speak and seeing the things that Dipty was talking about in these really beautiful and engaging graphics. And we had the great fortune of having this really dedicated and talented team of videographers and editors and illustrators and graphics designers and a suite of music that we could use to bring this all together for this whole experience. So for me, I think it was a behind the scenes picture, and like I got to be a major part of this effort to bring these fantastic tools together for a product that I really think will be valuable in the science classroom.
So for me, there's a lot that I learned through this. One is, I think I briefly touched upon this earlier, but I want to kind of circle back to it and emphasize it. As scientists, often we get several opportunities to talk to each other at conferences about our work, but we rarely get the opportunity to talk to the general audience about what we do. And I think and then that opportunity is great, but it also pushes us in thinking about how we can discuss our work to someone who is not a part of the community that we typically interact with. And that was something that I had to think a lot about in the making of this video, and learned a lot about over the course of this process too. And I think that's extremely valuable as a scientist to do. So yeah, that was one of the things that was great. And the other thing that I was kind of paying attention to as I was watching myself speak at that video is, I think it helps to some extent. There's a feedback process there. Once you know how to communicate a concept without using the jargon that we often lean upon as scientists, it crystallizes the concept more. So we go back to our work and we kind of infuse our work with some of the new things that we've discovered along the way. So I've also kind of, it's given me pause and made me reflect on my work differently. And that's all, taken our research in some different directions too, in different ways. I can touch upon that. One of them is kind of thinking about these eukaryotic signature proteins and what we can do in the lab to study them a little more. That's a direction that we weren't really pursuing, that we've started thinking more about as I've been thinking about the content for these videos. So yeah, those are two things that kind of come to mind right now.
Great, thank you. Dipti, we have a couple of questions about methane and methanogenesis. We have one asking if the methanogens that you study are part of the Loki branch or unrelated, and then also another participant's wondering about if most of the methane released is from Archaea, how do we reduce those emissions?
Yeah, both great questions. So the first one is about the Loki Archaea. No, the ones that we study in the lab right now are not a part of the Loki branch. They're a different branch of Archaea called the Eukaryotic that is slightly distantly related to these Lokis. And till date, I don't think methane metabolism has been identified in the Loki branch yet. That being said, the ability to produce methane in large part is the biological ability to produce methane in large part is kind of restricted to the Archaea. So this metabolism is something that is unique to this domain of life. And that kind of goes to the other question, which is, if most of the methane released is from Archaea, how do we reduce emissions? And that's at the heart of a lot of what, the work that we're thinking about. And one way to think about it is why do these organisms make methane? And the reason why they make methane is that it's coupled to the generation of energy in these organisms. And all of us, the reason why we eat food is to generate energy so that we can do all the things we do. And that's what these organisms are doing, is coupling the production of methane to the formation of energy. So if we can kind of using CRISPR tools, if we can kind of divert the production, if we can divert the production of methane to the production of something else that's less innocuous from the viewpoint of climate change, but still allow these cells to generate energy, that will allow these cells to survive and not kind of damage the ecosystems, most of which are natural in which they're found, but kind of attenuate the amount of methane that they produce at the same time. So those are kind of strategies that one can think about to reduce emissions from these organisms.
Great, thank you. Yeah, we've got another question from Esmeralda asking, what's the most difficult part of your research? And for Brittany, what's the most difficult part of your work?
I'll give you a break, Dipty, and I'll start. I think the most difficult part of my work is I work for an organization that is thinking about lots of interesting stories in all of life sciences, and choosing the interesting stories that haven't been covered elsewhere is truly a challenge. Like, there's infinite information out there, and we were so fortunate with this project. There's not a lot out there on Archaea, so we had kind of this whole wide open space to explore with this, and we're creating more things where we're exploring even more. But I do think it creates this kind of kid in the candy shop type feeling where there's so much interesting and fascinating things going on in biology, but at the same time, we don't want to duplicate really great resources that are out there, even if it's from a different organization. So kind of balancing our interests with where the needs are, I think, is probably the most challenging thing.
Well, for me, I think the most difficult part of the work that I do is also the part that I'm starting to enjoy a lot, which is, justifying why what I do is important and conveying its importance. It's not something, especially at this level, that where we're kind of communicating with a large audience of often non-scientists. We don't have to as scientists do that, but I think it's important, and it is difficult as scientists, but I think it's important, and it needs to be done. And that's something I'm getting more and more involved in, and I find that extremely useful and beneficial. So I would say right now that's probably it. And also, in terms of the research itself, I would say Archaea are understudied. I think that point was maybe very evident in the video. What that means is when you think about something like Archaea that a lot of people still in the biological domain as scientists don't study, it means that you kind of had to innovate constantly and come up with new ways of doing experiments. You encounter interesting challenges along the way. So you're often kind of flying by the seat of your pants in terms of, moving the research in interesting new directions, because often you're the first one doing something like that. So that's an interesting challenge of working with, a non-model organism is what we typically refer to these kinds of life forms in the lab. So that's always fun for me, but I guess people in my lab might not kind of agree with me sometimes there.
Great, thank you. I want to make sure we get to a question from Dulce. She asked if Archaea is the ancient bacteria and eubacteria is the new bacteria, then why are bacteria presented first in the tree of life imagery?
Okay, I'll field this, and Brittany, feel free to add something. So I think Archaea, the name, I think indicates that they're ancient, but that doesn't mean that Archaea are any older than bacteria. It's just that that was a name kind of ascribed to them, and in terms of kind of going back in time, I don't think right now we can say whether an organism that looked like a modern day Archaean or a modern day bacterium came first. It's really hard to make those inferences. But what I do want to kind of emphasize is that both groups of organisms exist today, and we shouldn't probably think about one being newer than the other, but that they're different and not closely related to each other. That's kind of the point that I was hoping to get across in the video.
Yeah, I think the only thing I can add to that is just thinking that and recognizing that they split off from each other, what, billions of years ago, right? And so their evolutionary kind of arcs have been distinct from each other for such a long time. And it is hard to trace back to kind of that chicken and egg of which one came first. But we know it's been billions of years of them being distinct from one another.
Great. We have an interesting question from Jim, who was wondering about you. In the video you talked about just sort of the serendipity of discovering Archaea and that proximity playing such a big role. He was wondering if you could speculate on if and when the distinction between Archaea and bacteria would have been discovered without that coincidence of the labs being right next to each other. I wonder if were there other groups that were maybe close, or do you think it would have taken longer? What are your thoughts?
I think it's safe to say that Carl Voss was definitely pioneering and championing the 16S ribosomal RNA. I would not say there was any kind of close competitor there. But history is played out the way it did. But if you would let me speculate for a bit here, I think if he hadn't received the sample from Ralph Wolf, he would have still kind of published his groundbreaking technique, which I do want to emphasize that technique in itself is groundbreaking. And I also do want to emphasize that Carl Voss didn't build the technique to look for Archaea. He was just trying to document how organisms are related at the level of molecules that exist inside the cells. So once that groundbreaking technique would have been documented, I think it was just a matter of time that someone would have, applied it to an archaeon and found the differences. So it might not have been him directly, but I think his technology was paramount for that discovery to be made. And it just so happened that he made those two discoveries simultaneously. And that's the part that's serendipity to me.
Yeah, I think there are a lot of examples in science of people that were having lunch together and just realized there was an overlap and we didn't, and something really amazing happened out of it. So having people, bright minds in proximity to one another is always a really good thing too.
Yeah, it's true that serendipity is a key part of science, and you'll never know what will bring two things together or multiple things together to lead to that foundational discovery. And in fact, that's a major theme of a video that will be kind of giving a pre-release screening of tomorrow at 4 o'clock Pacific time on the Messelson stall experiment. So if you like this idea of the chance events that lead to major scientific discoveries, that's a core component of that video that we're giving a screening of tomorrow as well.
All right. Let's see. There's another question earlier on somewhere about Dipti. Do you know anything about what the connection is between Norse gods and the names that were chosen, or why Norse gods were part of all that?
My recollection is that the first samples of the Asgard Archaea were taken from marine sediments. And that region where they sampled, I think was called Loki's Castle. So it's off the coast of one of the Scandinavian countries. I don't remember which one. And because it was called Loki's Castle, and then that's kind of where the first Asgard Archaean was found. Those were called Loki Archaea. And then, they just kind of built on that name. And now the rest of the group is kind of named after different Norse gods like Odin and Heimdall, Thor, and such.
Great. Another question just came in. Brittany, I wonder if you could talk a little bit about the visuals. They were really beautiful, and what was that design process like, taking information that needed to be conveyed and also making it really visually appealing?
Yeah, so thank you for that feedback. We're really happy and in love with how the graphics turned out. And there were multiple parts that came together for that. So we have a team member who is an artist, and a lot of those were hand drawings. They started off as hand drawings, And they exist on a resource called the Explorer's Guide to Biology, which is kind of a sister arm of the iBiology program here. And those illustrations served as a starting point of the inspiration for, well, if we're trying to explain the differences in DNA sequences as organisms evolve, and then looking back at those DNA sequences to make sense of how closely or not closely related they are, what are the things we need to show? Well, one part is DNA, and one part is different types of species, and another is looking at the DNA at the nucleotide level. What are the actual bases? And so I kind of put together what I thought could be shown when. I have zero skills in graphics, so it was really just me collecting the resources that I thought and writing down descriptions of what I thought could be conveyed. And then we actually pass it off to our graphics team. So we have, we work with 2 people who just have this immense skill set, and they kind of wove it into something that was so beautiful. And it's almost poetic. It's visually poetic in the end. And I think it just creates this feeling that you're immersed in it, and it feels incredibly beautiful, and it's beyond what I could have imagined. So I have to give credit to our artists and our graphics folks as well for putting that together. But yeah, it very much helps to have that expertise in biology to know what are the critical things, and then what can be kind of approximated using design. Kevin, I don't know if any other questions are coming in, but one point I kind of wanted to cover is, the strange world we're living in right now, where we're all sitting in our respective homes and connecting via the internet. And we debuted the video 2 weeks ago via a YouTube premiere. And Dipti, you had this wonderful anecdote of your parents tuning in from India, and how that was an opportunity for you to connect with them about your research. And do you want to share that? I thought that was just so great to hear in this strange time.
Yeah, sure. I think, yeah, there are these, these times are strange, and I would rather go back to the world the way it was. But I think there's small things to find joy in. And one of them was the fact that this video premiere happened online, and what that meant. So I sent the link to my parents. It's midnight. They stayed up, which they typically don't for anything that I know of. They stayed up for this, and they watched the video. And both of my parents my dad's an engineer, but neither of them had like a basic fundamental biology degree. And, it's, well, they kind of have this sense of what I do. I think watching the video kind of really helped crystallize these concepts that are often abstract to communicate, even sometimes to your parents, with them. And, I got they were super excited. They called me. We had this long conversation. And I think that was probably the first time that, I've kind of really gone down in the weeds with them about the controversies of, the work that I'm doing in terms of the 2 versus 3 domain tree of life, and how some of the stuff that I'm doing could potentially help, figure out what the topology of the tree of life is. So yeah, it was great. It's probably a conversation that wouldn't have happened if this video hadn't happened. So that's one of the things I'm really, really glad about.
Yeah, I really loved hearing that story from you. And I think those are the types of things that we hope these videos create. They create kind of space for conversations about what are the interesting questions that are happening in current biology, but also for the things that we know, what were the discoveries that got us there? And I think that this video captures those key experiments that happened, that Carl Bose did in the 1970s, but then what's changing today, and the fact that we only know as much as what our current evidence tells us in biology, and it's always open to changing. There's could be new evidence, and Dipti brought that up in the video itself, that in a week or 2 we might have new evidence that the tree doesn't look the way we currently think. It's going to be something else, and that's just how science works. And it's just a fascinating thing to be involved in and to learn about.
We have another question about how, for each of you, how do you think or hope that this video is going to be used in education? You both participate in education in different ways, from different angles of it. From each of your perspectives, what do you think the use of this video will be?
Well, I can go first, and I think it just builds on what Brittany and I we're talking about. I think when we teach in a traditional classroom setting, we often just convey facts, and we don't convey how we get to those facts. And we convey them as if, someone knew that this had to be done and they did it and they showed that it was right. We don't kind of walk through maybe often a slightly meandering process of science. We don't talk about the, serendipity in science. We don't talk about how people got there. And I think this kind of a video often kind of overlays the facts themselves with the story behind the facts. And I think that just only helps with the process of education. And I think it's a great way. I mean, I'm definitely going to try to see if I can use it for the class that I'll be teaching, because I think it's really, really useful. And I think it helps you remember these things better. That's my personal opinion.
Yeah, I think the key thing that Dipti, you just touched on is that this is conveying how scientific knowledge is accumulated and how it can change and what it's like to be doing science. And along the way, while you're kind of hearing about these things, you're learning that actually scientists are looking at the DNA level to make these links in evolution nowadays and what that actually looks like. And so we have the strands of DNA passing each other and aligning in certain ways. And I think driving those points home, there's a lot of components of biological systems, and it can end up being kind of an alphabet soup for students where there's just a lot of terms being thrown around and it feels like a lot of memorization and things that they're supposed to be learning without the context. And I think what this video and future videos that we hope to create will do is create that context of zeroing in on something where it's starting from a question perspective, the experiment that sought to answer that question, how it was done, how it was interpreted, and you learn kind of the facts along the way rather than starting with the facts first without that context. That's not engaging and it's not fun for students.
And just to let you know, we have a message from a tutor someone who tutors in the Bay Area, and they're planning to share this video with their supervisor and biology students. So it's making its way out into the world even more. So
that's wonderful. So we've been fielding a lot of questions, and I had promised at the beginning that we had a post-video poll. Let's do that now, and that can give some other folks some time to send in some questions for the remaining part of this. So I'll launch the poll. There's 3 questions in total, and we want to know who you are and what you thought. So take some time to fill that out. We'll run it for about a minute. So just some live updates. We'll run it for about 20 more seconds, but about 10 people are saying they learned something new. So we're happy to hear that. 60% of respondents said they like the music or graphics. We appreciate that too, because it's something that we think we're kind of carving out a new path with this. And 50% want to learn more, so stay tuned. Again, we're hoping to create many more resources around this fascinating area of research because it's just, there's a lot going on right now and new discoveries are being made every day. And we asked who you were. The majority of you tuning in live in the Bay Area. At least one person lives outside the United States, so thanks for joining in. If you're in a different time zone and it's late, we really appreciate you being here. Another person lives outside California. And then as far as who you are, we have some educators on the call. Looks like we have 3 educators, 3 scientists, 1 high school student, 1 college student. So a range of people, a parent and someone who's retired. So I'll end the poll right now. Let me share the results. So we thank you all for tuning in. And of course, we have this slated until 7:30, so we're happy to continue to field questions. But it's nice to kind of get a snapshot of who's tuning in and what you thought of everything. So largely the video was found to be interesting and you learned something new. And it seems like most people are tuning in from the Bay Area, which makes sense. This is the Bay Area Science Festival. So, thank you all for sharing your thoughts with us. So Kevin, are there any more questions coming in?
I do want to address a couple of questions that had been in previously. I think we have a few, and we have some high school students that are in the audience, and I wonder if both of you could just talk a little bit about, if you were a high school student right now, what kinds of things would you think about to like position yourself to pursue interests or learn more about what you might want to do in the future? If either of you have thoughts about that, I think that might be appreciated.
Yeah, I think if it's possible, if you identify an area of research that you're interested in, then very often those areas of research have societies that are affiliated with them, and you might want to reach out to them. They probably have outreach materials and lists of scientists that you can learn more about. Like for me as a high school student, I grew up in a small rural town. We didn't have a research university nearby. The closest research university was an hour and a half away. And that's why I didn't know that research was a thing that people did for a living, because it wasn't part of my community. And so I don't want to assume that people have a university that they can go to and reach out to scientists directly. But these discipline specific societies, I think, are probably a great starting point for learning about the who's who of the field, and then kind of getting a sense of people that you can reach out to or learn from. That might be a great starting point.
Yeah, I agree with that. And if you, I think the one other thing I would say, maybe that holds more true today than when I was a high school student, is, and there are resources like iBiology that you should obviously use. Biology is huge, and kind of finding areas within it if you're interested in biology, by just kind of going through these videos. These are all amazing. I mean, these are videos that I've seen of fabulous scientists who I often, love, haven't had a chance to go see a talk by in person. So I've seen the videos and they're just spectacular. So it's a great way to kind of, I think, I'm going to make a shameless plug for iBiology. It's a great way to know what's happening right now. I think one thing that I kind of go back in when I think about my high school education is textbooks that I learned from didn't probably have the state of the art. They were the textbooks I learned from. So I'm making my own case here. Lagged, contemporary science by a few years, often decades, depending on what you're studying. So, knowing what's happening in the field right now is important. And, right now we have all of these resources. So that's definitely something that's relatively easy to do if you have internet access, and all of these things are free. The other thing that I can say is right now, this might be unique to the times that we're in right now. There's a lot of stuff that's happening online, more than before. So all of these societies that Brittany mentioned, they have conferences that are online that anyone can attend. And so what that means is you can, you have more resources at your disposal to some extent. You can attend a conference often free of cost, just to see what it is like to be a scientist, and what kinds of things people are working on. And the other thing I want to say is, don't feel intimidated by scientists when they speak. If there's a word that's mentioned that you don't know, it's not that you should know it, and only then the talk would make sense to you. I came to biology as a grad student, and I came from engineering with very little knowledge of biology. It took me a while to catch up, but attending these talks and kind of going to them over and over again just made the process easier. So I would say, don't be afraid of jargon. As scientists, we should probably minimize their use, but especially in broader settings. But if there is a word that you don't understand, don't be dissuaded by it. Yeah.
And just one last, we appreciate, Dipti, your energy. Dipti is not a member of iBiology, and we appreciate your enthusiasm and sharing it as a resource. We have 600 videos, and going back to this idea of societies, a society that would be relevant to the topic we've been talking about tonight is the American Society of Microbiology. So if you have an idea of a topic that you're interested in, Googling something like the Society of something will help you navigate to those resources. There's typically one out there for every region, every subdiscipline, whether it's cancer or microbiology or molecular biology or something else.
Sort of along these similar lines, I wonder if you could touch on, you both serve as mentors in different capacities as well now in your careers, but I wonder if you could talk a little bit about mentors that were important for you and how they played a role in the progress of your paths.
Yeah, I can start. I had a mentor in college who was really the key person to get me on my path to doing science and being a researcher. And it was because we shared a similar background. We were both first generation to college. We both liked things that didn't seem interesting to other people who were interested in science, like music and culture and things. And of course, that's a vast oversimplification, but as a first generation college student coming to college and not knowing much about scientific research, I really felt on the outside of it when I was talking to my professors and feeling like, as Dipti was saying, I didn't have the vocabulary. And this professor made me feel like I could connect and do it and put her faith in me. And so that was the first mentor that made a huge difference for me. And I had mentors that I did research with in college, and they were key for helping me recognize like how to navigate the system. There's this idea of this hidden curriculum that exists for academia, and it's very true. If you aren't told how to design your CV or how to write a letter or write an email to somebody, you might not get it right the first time because there's a set of expectations. And so I had mentors that helped me kind of navigate that. And I think that was super valuable to me. And what I can say is, if you find somebody that takes an interest in you from an academic perspective or a learning perspective, that person can help you draft emails to other people or navigate certain like fellowship applications. Those are all things that you can ask of someone if they want to help you get to somewhere. And I think it's not just about tests and academic records. It's really about navigating a whole system that exists that some people don't innately know how to do. And I was one of them. And then for graduate school, I was fortunate to find a mentor who allowed me to pursue my interests. And so it wasn't that I came to a lab and my project was set up for me. We proposed a project, and after a few months, I realized like that's just not where my heart is. This is the thing I want to do, and he was completely supportive of it. And I was able to craft a thesis research project that I was totally invested in and totally interested in. And he totally let me kind of devise my own path, but of course provided me guidance along the way. And I hugely appreciate that too. So those were kind of the major players for me.
I mostly have to echo a lot of what Brittany said. I'm new, as I said, I moved from engineering to biology, which was a pretty big shift for me. And, you know, I think since that kind of shift in academic disciplines, it's kind of been working with two amazing people, my PhD advisor and my postdoc mentor. That's kind of really led me to where I am today. Like, if it weren't for these two individuals and a few others along the way, you know, there's so many small things. The hidden curriculum, that's a great term. There's so many small things along the way that, you know, if you don't have someone who guides you on how to do these things, I mean, as simple as writing an email or knowing how to write the first draft of your first paper ever, you need someone who kind of is willing to sit with you, you know, teach you how to fix the mistakes that, at least for me, I made plenty of them, and then learn from them as you move forward. That was critical. So yeah, I mentorship is extremely valuable. And as someone who is, you know, a new assistant professor, and I'm kind of growing my own lab, I've kind of switched roles a little bit. Now I'm in the position of being a mentor, and I have several mentees in my lab. So it's that information transfer that is also a big part of my job now.
Great, thank you. Thanks, both of you. We have another question. Maybe Dipti, you can start, and we can start wrapping up beyond this. But yeah, I guess we were just curious, what is next for each of you?
Well, I'll just circle back again to what I just said. I started my own lab as an assistant professor at UC Berkeley a year ago, slightly more than a year ago. So what that means is I'm recruiting a group of people to my lab. We have space and we're kind of taking off from where I left my postdoc, which is what we typically do in biology after we finish our PhD before we get faculty positions, if that's the route we want to go down. And what that means is what's next for me is building a research group, I have graduate students, postdoctoral fellows, undergrads in my lab. We're all kind of fascinated by different aspects of these methanogenic archaea. So kind of building the momentum on that is probably what's next.
And Brittany, what about you?
Yeah, so what's next? I think we have kind of created this new model of the potential for iBiology videos. And for those of you that maybe haven't gone to our website or seen our videos before, this video that we showed you tonight is actually quite a different flavor than what we have created in the past. So we have 600 something videos in our library, and largely what those are a scientific seminar format where a scientist is standing in front of their presentation and talking through their research and talking through their slides. And as you saw tonight, what Dipty was doing and the images we were showing was a very different kind of formula. And this is something that we want to explore more on. And so for me in the near future, it will be identifying new topics for which there aren't educational videos out there, and thinking from the lens of educators, what can we create that tells about the process of science, tells real life research stories and current research stories, and gets people excited about, learning new things and collecting evidence and changing your mind based on where the evidence is taking you.
Let's see. Dipti, you were touching on this a little bit recently and then before we got on the call as well, but I wonder if you could also just talk a little bit about what it's like to be a researcher right now and how you're adapting to sort of the strange situation we're all in.
Yeah, right. So I first want to give a sense of what it means to do research, which is that a lot of my work is what qualifies in biology as a wet lab, which means that we physically go to a lab space where we, have chemicals and reagents and a bunch of specialized equipment that we need to use to do our studies. And since the pandemic, for the first few months, we were obviously not allowed to go back in, but now we've been back in lab. We have several restrictions that have been put in place to make sure that research happens safely. So what that means is everyone in my lab, we can't all be in at the same time. So a few of us have to be in at once. So we're kind of taking shifts. Lab meetings can't happen in person. So a lot of, like this webinar, a lot of our interactions are kind of transitioning online through some format or the other. So yeah, I think we're in lab, we're doing work, but we're obviously taking into account all of the different restrictions to keep us safe. And the thing that I think I miss the most is just face-to-face interactions, meeting someone in the hallway. But I hope in the next few months those kinds of encounters will resume in some way, shape, or form. The one thing about this time that I think as scientists is interesting is one thing I can mention from my own experiences. I was recently invited to give a seminar at another school in a different state. And under normal circumstances, I would have gone there physically and had a day long meetings and then given the seminar and come back. This time, my lab got to participate in this seminar with me. So it was kind of like taking my whole lab there with me, which is an experience that we wouldn't have. So in some way, we're including more scientists in the mix, but in other ways, we're kind of, because of the restrictions, we're a little more isolated.
And Dipti, a question I have, and I don't recall that we covered this so much, but could you paint a picture, outside of the pandemic scenario, what is it like to be in your lab and doing your research? What types of equipment are you working with? How do you actually culture your microbes? These are all things that I'm sure people would love to hear about.
Yeah. So one thing that I think wasn't covered in the video is that these methane producing archaea, the methanogens, they have this one unique quality to them that they don't like oxygen. They, in fact, are only found in places that are devoid of oxygen. Our atmosphere has about 21% oxygen gas, so we have to exclude that in everything we do. So what we have are these chambers in our lab which we fill with gas that is completely devoid of oxygen, and it is only within these chambers that we have to grow these microbes. If you have to envision these chambers, think of a giant inflatable balloon, if you will, that's filled up with, nitrogen gas instead of the atmosphere that we breathe. So that's kind of a big part of our day-to-day life, is working inside these chambers to keep our methane producers happy. So yeah, and then besides that, it's kind of a standard molecular biology lab. So we have kind of instruments to do specific genetic techniques in the lab and such.
And another cool thing is, of course, I learned this from the interview, and it wasn't revealed in the video that we just shared, but it will be in subsequent videos, is the microbes that you're studying, kind of the ones that you've been focusing on for most of your work with Archaea, come from a pretty interesting location. And do you want to tell us about where they were discovered and when that was?
So the microbes that I study are also native to California. And so they don't come from too far away. The specific ones that I study right now come from San Diego, so specifically La Jolla. And they were kind of found in something that's called a kelp forest. So lots of kelp growing kind of just under the surface of the ocean. People saw these giant bubbles coming out of these kelp forests. So once someone went scuba diving and they were a part of someone's lab, so they just kind of took this, essentially a microbial mat is what you would call it, within the kelp forest, and brought it back and tried to isolate what was living there. And one of the things that was living in that mat was a methane producing methanogen, this archaea that we study called Methanosarcina, acetoborans. So yeah, again, talking about serendipity in science, I don't think someone went there looking for methanogenic archaea. They just happened to find one.
And I think one thing that's interesting too is that, there's so many millions of microbes that exist in the world, and very few of them are able to be kind of harnessed and taken into the lab and live in the way that they typically would. And so, there's probably so many more living in that kelp forest, but it happened to be that one was the one that survived the culturing procedure. And I think that's something that is interesting. I think biology is moving in a direction where we're developing the tools to both profile microbes in the space that they live, but then for those that we can bring back to the lab, we have really great tools for looking them at even at the single cell level. So we're moving away from this requirement that we have to have a continuous culture of cells, because that often eliminates some of these ones that just for whatever reason, we don't know the way to keep them happy and keep them growing. So I think we're in an age right now of microbiology where we have tools where we can dive into so much more about the microbial world and learn about it without being limited to the ones that end up surviving in our labs.
All right. Well, we are approaching the end of our time, but I do, if there are any other questions, feel free to send them in. Maybe Brittany and Dipti, do you want you have any sort of final thoughts as we wrap things up about the project and just sort of your own reflections on how this has come together?
Yeah, sure, I can start. The one thing I want to note is I was very impressed by the poll that Brittany sent before we showed the video and how many people in the audience already knew what archaea were. So that was great. I was very pleasantly surprised by that. And I've watched the video before, but in watching the video again with all of you, the world of archaea is great. The fact that I had this amazing opportunity to work with Brittany and kind of showcase, the work that keeps me and members of my lab excited on a daily basis, kind of showcase that to a much broader audience. It makes me feel really happy. So yeah, that's kind of the note that I want to end this on. I hope people in the audience some of whom may have left by now, kind of got to see some of the excitement that about this field and what kind of keeps us going.
Yeah, and I think the note that I'll end on is I think we are on the cusp of maybe an awareness outside of the microbiology world of archaea becoming more mainstream. And I remember a conversation we had on the day of your interview, Dipty, is that, there will be news articles about discoveries related to methanogens, and they'll be calling methanogens bacteria. And that's just categorically incorrect because there aren't methanogens that are bacteria, they're all archaea. And so up to very recently, there has been just a misunderstanding of what single-celled microbes are. They've been equated with bacteria, and that's not the case. And we know that archaea are this fascinating and unique class. And I think there will be an emergence of more information about them in kind of mainstream news and awareness of them. And I'm excited to see that. And I think this video is one piece of that puzzle, particularly being used in classrooms for that. So it seems like there aren't any more questions coming in, and I'm perfectly okay with ending a few minutes early on a Friday night. So I want to end by thank you all for tuning in, especially for those of you that hung on to the very end with us. It's been wonderful to connect, and we hope that we can do more projects like these and share them in the Bay Area Science Festival when we go back to being in person, because those are also really fun events. But thank you to the organizers of the Bay Area Science Festival for making this possible and very simple and easy. And thanks Dipty and Kevin for joining tonight and just engaging in this conversation. It's been really fun.
Yeah, thank you.
Okay, have a good weekend all and stay safe out there and take care.